Hi, my name is Adam. I am a top MCAT scorer and MCAT tutor here at Shemmassian Academic Consulting. And today we're going to be walking through a biochemistry passage, and I'm going to be showing you some tips and tricks that you can apply to your own passage analysis, and also just how a top scorer would be walking through an example practice passage like this. So let's jump right into it. Okay, so this is the passage that we have today. It's a biochemistry passage. To give you a little bit of context about where this is going to show up, this would be in the third section of the MCAT, meaning that we've just taken Chem Phys and CARS, the critical analysis and reading section, and the chemistry and physics. We've done the first half of the exam. We just had lunch. Now we are coming out from lunch and we're in this biochemistry biology section. That's where you would see a passage like this. So that's a little bit of the context. My strategy that I take when I'm approaching a passage like this is to look at the questions first, get an idea for what I'm looking for, then go over into the passage, try to pick out that information that I'm looking for from the questions. There may even be an opportunity that I can answer a few of the questions before looking at the passage. I'm not sure if this is going to happen yet with this passage, but it's always a possibility. And I'm looking for that because some questions, even though they're phrased in the terms of being part of a passage, can be answered without the passage. So let's take a peek at these questions here for this passage and see what we need to pick out here. Okay, so based on figure 1, which of the following conclusions is most likely to be correct? Okay, so this is definitely going to be passage based. We're going to have to peek at figure 1 for this one. Number 3, a student identifies an active site mutation in the HMG-A protein leading to its inactivation. Which of the following is the most likely mutation? Okay, so we have an active site mutation in this particular protein that leads to inactivation. Okay, we can answer this. We might be able, well, we should double check to make sure that there isn't anything going on with the active site odd in the passage before we answer this, but I'm thinking this one we might need to answer. We might be able to answer without information from the passage, because we know in order to alter an active site and make it to go from active to inactive, we have to see a change. Like that, something different has to be going on. So I'm going to peek at these really quick, just as long as we're on the questions and I'm thinking about it, so as not to lose any time here. So let's look here. This first one is alanine to valine, A to V. Those are both going to be the same properties. So those are the same properties. So we're seeing no change. So therefore that's not going to make it go from active to inactive. So that's going to be out. Then we see E to D. That's negatively charged to negatively charged. That's out. We're seeing no change there either. So I'm starting to see a trend here. We're seeing no change, no change. I'm guessing out of these last two, one of these is going to be making a change and the other one is not. Okay, Q to N. Q is a polar, uncharged. And N is also polar uncharged. So we're going polar uncharged to polar uncharged. So that would not make any sense to make it go from active to inactive, so that one's out. So I'm already going to answer D. I haven't even looked at it yet, but just guessing it's going to be a change. Okay, and we're seeing R to Q. R is positively charged. So we got a positively charged amino acid to Q, which we already mentioned. That's a polar uncharged amino acid. So that's definitely our answer. So that was a pretty easy one. We didn't even have to read the passage. All we need to do is know a property or the basic properties of our amino acids in order to get that one correct. Pretty easy. That's kind of nice. We already got a point. We haven't even had to read the passage yet. I appreciate that. The information in the passage suggests that which of the following molecules produces the brown pigment. Okay, we're definitely going to have to figure this one out from the passage because we're talking about information in the passage. We're looking particularly for the molecule that produces brown pigment. Okay, so I'm going to be looking out for that. Which of the following best described the Michaelis-Menten plot if HMGA if it shows cooperativity? Okay, this one I might want to peek at HMGA. If HMGA is really odd, I might want to peek at it, but I'm guessing it's not, because they're telling us if it shows cooperativity, so they're not asking us about what HMGA is functioning in the passage, they're asking about if it's cooperative. So anything that's cooperative, our graphs are always going to look the same. We take our Michaelis-Menten graph like this, and our graph is going to look like this almost every time. And this type of graph that looks in this point, that's not hyperbolic, that's not quite quadratic, that's a sigmoidal graph. It's not exponential. So that one we can also answer pretty easily just by having an understanding of Michaelis-Menten kinetics and how we can plot graphs and what cooperativity looks like on a graph. And it's going to always be sigmoidal, so that's pretty easy. Okay, so this is great. We had 5 questions. Just by starting with the questions and approaching them, we were able to answer 2 of them without reading the passage yet. So that's kind of nice. Now, as we go back to read the passage, the information we're looking for is the molecules that produce the brown pigment, homogentizate 1,2-dioxygenase mRNA, and how that's associated with this. The fact that they're adding it in, so maybe like it gets removed at some point, now they're adding it back, they're asking what's going to happen. And then we're looking at figure 1 and the conclusion from figure 1. So this is not that much information that we actually need to pull out from this passage. So let's go over to the passage and see if we can just pull out this information and get these questions right. Yeah, in general, when we're looking at passages like these, we don't need to understand everything in it in order to answer our questions correctly. There's probably going to be a lot of stuff in here that's more like a graduate level knowledge or above our heads. The MCAT's never going to ask you about that type of stuff. So why spend time on it, right? So don't waste your time. This is a timed test. If we all had unlimited time for this test, we would have no problems. All of us would get, 528s. Like, there would be no differentiation. So we need to make sure that we're not only being accurate, but we're doing it very efficiently, doing it quickly as well, because it is a time test. So information in passages that we don't need to digest to get us actual points on the exam, such as these 3 questions that we have here, should be going in one ear and out the other. Okay, so looking here at the top of the passage, Vibrio angularum 531A is isolated from deceased fish. Diseased fish in the Atlantic Ocean is a mixture composed of about 95% and 5% of highly pigmented cells. Okay, so we have highly pigmented cells, which is the 531AD, and then normal pigmentation, which is 531AC. Okay, so we're kind of getting set up. I remember one of the questions asking me about this, so I'm going to pay attention to it's highly pigmented, shows up 95% of the time. I'm not sure if that's important yet, but that's what we're looking at here. Okay, now we're going forward. Analysis of B angularum 531AD. Okay, we're still talking about 531AD. DNA region encompassing genes involved in the tyrosine metabolism showed a 410 base pair duplication within the HMGA gene that results in a frame shift and early termination of translation of the homogentizate 1,2-dioxygenase. Okay, that's a lot. That is a heavy sentence right there. But what can we pull out? We can pull out homogentizate 1,2-dioxygenase, because that's from our questions. And we can pull out early termination of translation. That, I remember there being something about mRNA of this, and so that's related to translation. So we'll look more specifically about what that question's actually answering, or actually asking us. But we know that this 531AD is causing a frame shift. I don't remember any questions about mutations, but it doesn't hurt to know which type of mutation it is, which is causing this early termination of translation. That's probably going to help us answer that one question. We'll look at that later. Researchers hypothesized that this mutation results in accumulation of homogentizate that is oxidized and polymerized to produce pyomelanin. Okay, so this accumulation of this, the mutation results in this that is oxidized in polymerase to produce this. Okay, so it's just that's just telling us the steps there. I'm not going to spend any more time on that. I'm not sure if that's helpful yet. To investigate the origins. Okay, now we're looking at the origins. So the origins of these two strains that they've already mentioned, researchers cultured them separately, of course. First, they found that 531AD was brown and 531AC was clear. Okay, I remember something about brown pigmentation. So I'm going to highlight that. 531AT, brown. I'll also highlight 531AC, clear, just in case in one of the answer choices there's something about clearness. Then researchers carried out whole genome sequencing. Okay, good for them. In both of the cultures, researchers found 23 different mutations, including the HMGA mutation. Good. They also identified a Q349P mutation in the tyrosine-specific transport protein coding sequence. I don't remember any questions on this, but it might be involved in the answer choices, so I'll highlight this just to know we're talking about it. And I'm also going to highlight that this is what that is. TYRP. Software folding simulations did not show structural changes in the protein. Okay, so this mutation didn't cause structural changes, leading researchers to believe that the amino acid change does not affect tyrosine uptake. Okay, so this whole thing and the software that they used is suggesting that there's no change. So I'm just going to say, I'm going to go back to this mutation because this whole end part's about this mutation. The mutation in this sequence does not affect anything. That's what the passage is telling me there. Next, researchers wanted to ensure that the strains were derived from each other. Okay? From one another. So I'm going to highlight them, and I'm going to say from one another. So what did they do? They harbored a PJM1 plasmid, of which most strains slightly differ, and endonuclease digestion was carried out to determine whether or not restriction patterns between 531AD and 531AC were identical. Okay, so they basically did endonuclease digestion and then looked at restriction patterns. I don't know the details of endonuclease digestion, but I don't think any questions would ask us about that. Alright, we don't have any on it right now. But this, so they're kind of setting us up for this figure here, right? Because now we have figure 1, which is showing us that, yeah, okay, the restriction patterns. We're seeing those restriction patterns. So we're looking at this description of the figure. We're seeing restriction patterns of the two genes that we care about, 531AD and 531AC, and they're labeled as PJHC1D and PJHC1C. Okay, and as we look at, let's zoom in a little bit further here, it's pretty small. We look at these graphs, or sorry, at the figure here, we're really just going to want to compare these, right? Well, it's all we care of whether or not these are identical to each other. I'm not really caring that much about PJM1. But if we look at these, it looks like first line, they're lining up. It looks like it's looking pretty identical across the board on the way down. It's like if we're comparing the kilo base pairs on the A there, and then as we compare this restriction, okay, the 1D and 1C looking very similar. Okay, so if their purpose was to see whether or not, right, their purpose was to define whether or not 531AD and 531AC was derived from one another, most likely this figure is going to say yes, they are, because they're looking almost identical. Like if you were to switch the labels for these, nobody would notice. That probably means that they're derived from the same genetic source or in some way similar. Okay, now let's read this last paragraph here. In a final study, researchers complemented strain 531 with recombinant clone harboring HMGA. So we got complemented this with that. This restored the original color, confirming that in the absence of this, so that's okay. So that's the They're just straight up just going to tell me the conclusion. In the absence of that, homogentisate accumulates and undergoes non-enzymatic oxidation and polymerization, resulting in high amounts of brown pigment. So even in the absence of that, we still have high amounts of brown pigment. Okay, that's kind of helpful. Still talking about brown pigment. I know there's a question about that, so good. I'm feeling pretty good on the passage there. I feel like I've highlighted important things in case I need to refer back. And let's answer our last 3 questions here. See how they go. Based on figure 1, which of the following conclusions is most likely to be correct? Okay. Based on our quick analysis of figure 1, the conclusion that I picked up was that they're the same. And that's what they were trying to do, is like, are they the same? So I'm looking for, do they come from the same thing, or are they the same? A, the strains come from different origins. Okay, that's the opposite of what figure 1 says. B, the 2 strains are derived from a common origin. That seems good. We're going to peek at C and D. They have to be really good to be better than B, but let's see. The 531AC strain was taken up via transformation. I don't think figure 1 was saying when the strain was taken up. That's not based on figure 1. So this might be true. It could be a true statement, but it wouldn't be answering the question that's being asked. They're asking the question about the conclusion on figure 1, so I'm gonna rule out C there. And then D, early V. angularum produced strain 531AD first, which then mutated into strain 531AC. Well, that's possible, but that's not what figure 1's talking about. This could be true. I'm not going to deny that it could be true, but that's not what the passage or the figure was talking about which one came first, or taken up via transformation, or any of that stuff. That's not what figure 1 is discussing. It's just whether or not they were from a common origin or not. So B, that's got to be the correct answer there. Okay, number 2 here. Researchers add exogenous homogentisate 1,2-dioxygenase mRNA to a culture of strain 531AD. Which of the following is the most likely result? Okay, so let's peek over. So mRNA to this, we know from the discussion on this in 531AD that this results in a frame shift that causes early termination of translation. So if we introduce the mRNA, how is that going to impact translation? Well, mRNA is produced from DNA, so that's actually transcription. So adding in exogenous mRNA would be to increase the transcription. The only thing that's going on here is a frame shift is causing early termination of translation. Okay, so this is kind of a central dogma question. So this is kind of like leading me to think that we're talking about from DNA to RNA to protein, and asking, do you understand where each step comes in, right? So we have transcription is this arrow, this is transcription, and this is translation. And they're asking, okay, if we increase transcription and we throw in more RNA, so we increase transcription, so we're just pumping up the RNA, basically exogenously impacting our RNA rates. How is that going to impact things? So the thing is that wasn't impacted before. Our RNA value, that was not the limiting factor. The limiting factor was early termination of translation. So the impact on translation was not affected. So I'm going to say that there's going to be no changes here. Let's see what our question choice is, which one leads towards no changes. The brown pigment remains. Okay, that suggests no changes. The clear pigment remains, also suggests no changes. The brown pigment turns into a clear pigment over time. That suggests a change. I'm going to rule that one out. And then the clear pigment turns into a brown pigment. That's also a change. Ruling out C and D right away. So now it's just, is it a brown pigment staying or a clear pigment staying? Okay, we're talking about 531AD. Pretty sure that's brown. I'm going to double confirm that. First, they found that a 531 AD had brown, 531 AC had clear. Okay, so it is brown. Okay, so that's going to be A, brown pigment. Okay, good. We already answered number 3. This is the change. The rest of them, polar charged, polar uncharged, negative, negative. Those are the same properties. They're aliphatic over there. Now number 4. The information in the passage suggests that which of the following molecules produces the brown pigment? Okay. 531 AD, 531 AC, pyomelanin, or homogentisate. Okay, well, it's not going to be... Well, first of all, 531 AC is clear. 531 AD is brown. It's not producing the brown. That doesn't make any sense. So I'm going to rule out both A and B. Okay, now it's just, is it homogentisate or is it pyomelanin? I remember highlighting pyomelanin at the end of that first paragraph. So let's go and peek at that really quick and look at that sentence. Mutation results, homogentisate, produce pyomelanin. So they're just looking at my highlights. Homogentisate produces pyomelanin. And then the strain 531 AD is brown. So homogentisate wouldn't be producing the brown color. The homogentisate would be producing the pyomelanin, which is producing the brown color. Okay, so the causal pathway is leading me towards pyomelanin over homogentisate. So I'm going to rule out that, and I'm going to answer pyomelanin. And we already answered this one. Cooperativity in Michaelis-Menten with anything is going to lead us to sigmoidal. Okay, I'd say that was a pretty efficient way of going through that, and I think we nailed it. That felt pretty good. Yeah, feel free to put any questions about anything I did there down in the comments below, and we will definitely answer any questions you guys have. So that pretty much wraps it up. That's my analysis of a biochemistry passage. I hope you enjoyed that. If that walkthrough was helpful for you at all or in any way enjoyable to watch, feel free to leave us a like, subscribe to our channel, and a couple other items you might want to check out. We have a YouTube playlist on different strategies to a 528. Check that out in our channel. We also have daily passages that can be sent to your inbox. The link for that is in the description, so passages just like the one that I went through today. So definitely sign up for that, and if you do, you're not missing out on any of these free materials that we can provide for you here at Shemmassian to give you the tools to get your own 528. So best of luck to you, and see you guys in the next one.